Information moves through the nervous system via two integrated forms of communication, which is electrical neurotransmission and chemical neurotransmission, as shown on the screen. The adult CNS contains over 100 trillion synapses, and most neurons release more than one neurotransmitter when stimulated. As in the autonomic nervous system, chemical transmission in the CNS appears to be particularly drug susceptible. Therefore, an understanding of the neurotransmitters involved is basic to an understanding of drug mechanisms. Essentially, these are the learning objectives. By the end of this video, you should be able to identify the major CNS neurotransmitter and describe its major receptor subtypes. And of course, you should be able to discuss the role of CNS neurotransmitters in modulating or controlling your brain function. An action potential is generated at the origin of the axon following sufficient excitatory stimulation of the neuron. The action potential is created by movement of electrically charged particles, which is ion, in and out of the neuron through pores, which are the ion channels, in the cell membrane in a particular pattern traveling along the length of the axon. When the action potential reaches the axon terminal, it stimulates the release of chemical neurotransmitter. After the action potential has induced the release of chemical neurotransmitters, the neurotransmitters pass information to the next neuron. The presynaptic neurons uses chemical signals, or rather neurotransmitter, to increase or decrease the generation of action potential in the postsynaptic neuron. It is also possible to affect other biochemical processes such as cell signaling pathways in the postsynaptic neuron. This complex interconnectivity gives rise to the rich functional communication network that is the central nervous system. The process of chemical neurotransmission is shown on the screen. Briefly, an action potential arrives at the terminal end of the presynaptic neuron, which means that the membrane potential at the terminal is altered. This altered potential causes voltage-gated ion channels to open, leading to a rapid influx of calcium. The increased calcium within the neuron terminal causes vesicles to fuse with the membrane and release neurotransmitters into the synaptic cleft. The neurotransmitters will then bind to specific receptors on the postsynaptic neuron. The effect of this binding varies, but it usually leads to altered conductance of postsynaptic neurons and the propagation of action potential. Neurotransmitters can either bind to ion channels, in which case they have a direct and immediate effect on postsynaptic function, or they can bind to G-protein coupled receptors, which results in an indirect and slower effect. These are the major CNS neurotransmitters that I will be talking about in the next few minutes. Essentially, we're going to look at the role of these various neurotransmitters in regulating brain function. Neurotransmitters can be classified based on their function. Basically, a neurotransmitter influences a neuron in one of three ways: excitatory, inhibitory, or modulatory. Excitatory neurotransmitters increase electrical excitability on the postsynaptic side through modulation of transmembrane ion flow to facilitate transmission of an action potential. Inhibitory neurotransmitter decrease electrical excitability on the postsynaptic side to prevent the propagation of an action potential. Whether a neurotransmitter is excitatory or inhibitory depends on the receptor it binds to. Additionally, there's also the neuromodulators, and neuromodulators function to alter the strength of transmission between neurons by affecting the amount of neurotransmitter that is produced and released. Neuromodulators are a bit different, as they are not restricted to the synaptic cleft between two neurons, and so they can affect large numbers of neurons at once. Neuromodulators therefore regulate populations of neurons while also operating over a slower time course than excitatory and inhibitory neurotransmitters. These are some of the differences between excitatory and inhibitory neurotransmitters. Essentially, the control of the central nervous system can be thought of in terms of a balance between the excitatory and inhibitory signals. If the sum of the excitatory signals that a neuron receives outweighs the sum of the inhibitory signals, then that neuron will exist in an active state and will continue to signal to downstream neuron, and vice versa. Examples of excitatory neurotransmitters include glutamate, acetylcholine, epinephrine, and norepinephrine, whereas examples of inhibitory neurotransmitters include Gaba, glycine, serotonin, and dopamine. But again, the excitatory and inhibitory effect is also dependent on the receptors whereupon the neurotransmitters act on. The major neurotransmitters and their receptors are listed on the table on the screen. The variety of neurotransmitters that operate within the CNS adds to the functional complexity of the brain. Different neurotransmitters can produce different responses on the same neuron. For instance, a neuron might be excited by serotonin but inhibited by the binding of Gaba. Defects in many neurotransmitter pathways have been implicated in CNS disorders. The chemical transmission in the CNS appears to be drug susceptible, as I mentioned earlier. Therefore, an understanding of the neurotransmitters involved in these diseases is the basic to an understanding of drugs' mechanism of actions. Let us first start with the role of acetylcholine in the central nervous system. Acetylcholine was actually the first neurotransmitter discovered and is named for the two substances used to synthesize it, which is the nutrient choline and the enzyme acetylcoenzyme A. Neurons that contain acetylcholine are called cholinergic neurons, and there are several clusters of cholinergic neurons throughout the brain. Some are found in the basal forebrain, and they include the medial septal nucleus and the nucleus of the diagonal band, as well as the nucleus basalis. Others are found in the brainstem, including the pedunculopontine nucleus and lateral-dorsal tegmental nucleus. Acetylcholine acts on two families of receptors, and each receptor family has several subtypes. One family is ionotropic, and they are called the nicotinic acetylcholine receptor because nicotine also binds to and activates the receptor. Their activation generally results in excitation of neuron. Another family is metabotropic, and these are called muscarinic acetylcholine receptors. Acetylcholine has a variety of functions in the nervous system, but in the central nervous system, its role is primarily excitatory, and it plays a role in arousal, memory, learning, and neuroplasticity. Another major neurotransmitter in the CNS is norepinephrine, also known as noradrenaline. Norepinephrine is a monoamine neurotransmitter, a term that refers to its chemical structure and the fact that it's derived from an amino acid. It is also a catecholamine, a term that again refers to its chemical structure and the fact that it contains a catechol nucleus. Norepinephrine also functions as a hormone. It is synthesized from dopamine in a reaction catalyzed by the enzyme dopamine beta hydroxylase. Norepinephrine-producing neurons in the central nervous system are primarily concentrated in the pons and medulla. The most prominent of these groups of neurons is a nucleus called the locus coeruleus, which is the main site of norepinephrine production for the central nervous system. Norepinephrine acts on G-protein-coupled receptors referred to as adrenergic receptors or adrenoceptors. And like any neurotransmitter, the actions of norepinephrine depends on the type of the receptor it activates and where that receptor is located. Although norepinephrine in the central nervous system is frequently associated with arousal, alertness, and attention, the full extent of its actions are more complex. Dopamine is a monoamine neurotransmitter that plays one of the major roles in the central nervous system. To synthesize dopamine, the amino acid tyrosine is converted to L-dopa, then L-dopa is decarboxylated to form dopamine. Dopamine acts on G-protein coupled receptors, and there are at least 5 subtypes of dopamine receptors. There are several areas of the brain where dopamine neurons are concentrated. The largest are the substantia nigra and the ventral tegmental area in the midbrain. Other areas include the hypothalamus, olfactory bulb, and retina. There are actually 4 major dopamine pathways that carry dopamine from these areas of concentration to other parts of the brain. The first one is the Mesocortical Pathway, labeled in purple, and this transmits dopamine from the ventral tegmental area to the frontal cortex. The Mesocortical Pathway is involved in cognition, memory, attention, emotional behavior, and learning. The second pathway is the Mesolimbic Pathway, labeled in green. The pathway begins in the ventral tegmental area and connects to the limbic system via the nucleus accumbens, the amygdala, and the hippocampus. The Mesolimbic pathway is associated with pleasure and reward-seeking behaviors, addiction, emotion, as well as perception. The third main pathway is the nigrostriatal pathway in turquoise. This pathway transmits dopamine from the substantia nigra to the striatum, and this pathway is associated with motor control, movement, as well as sensory stimuli. Last but not least is the tuberoinfundibular pathway in cerulean blue. This pathway transmits dopamine from the hypothalamus to the pituitary gland, and it influences the secretion of certain hormones including prolactin. Let us now move on to the role of serotonin in the central nervous system. Serotonin, or 5-hydroxytryptamine, is a well-known neurotransmitter that plays vital roles in neural activities and has been found to be intimately involved in emotion and mood. It is a monoamine neurotransmitter, and to synthesize serotonin, the amino acid tryptophan is converted to 5-hydroxytryptophan, and then 5-hydroxytryptophan is converted to serotonin, or 5-hydroxytryptamine. Serotonin neurons are primarily found in the brainstem in clusters of neurons called the rough nuclei. Serotonin neurons from the rough nuclei project throughout the brainstem and brain and provide serotonin to the rest of the central nervous system. Neurons that use serotonin is involved in functions including sleep, memory, appetite, and mood. Researchers have identified 7 different families of serotonin receptors which differ from one another in terms of their distribution, the substances that bind to them, and the effects they mediate. All but one of this family of receptor consists of G-protein coupled receptors, and the other receptor family, which is the 5-HT3 subtype, consists of ligand-gated ion channels. Within these 7 families of receptors, 14 receptor subtypes have been identified as well. Let's move on to the next neurotransmitter, which is the main excitatory neurotransmitter in the CNS, glutamate. Glutamate generally has excitatory actions, meaning that when it interacts with the receptors of a neuron, it makes that neuron more likely to fire an action potential. It is in fact used at the vast majority of excitatory connections in the brain and at more than half of all synapses in the brain. Glutamate interacts with several different types of receptors. There are 3 identified ionotropic glutamate receptors, namely NMDA, AMPA, and kainate receptors. When activated, all the 3 receptors allow positively charged sodium ions to flow into a postsynaptic neuron, depolarizing the neuron and making it more likely to fire an action potential. And then there's also the metabotropic glutamate receptors, and these receptors have more varied effects as compared to the ionotropic glutamate receptors, and they may be involved with excitatory or inhibitory actions. Glutamate essentially takes part in the regulation of general excitability of the central nervous system, in learning processes, and memory. Last but not least is Gaba, which is the main inhibitory neurotransmitter found in the brain. In other words, when Gaba interacts with the receptors of a neuron, it generally makes the neuron less likely to fire an action potential or release neurotransmitter. GABA is synthesized predominantly from glutamate by the enzyme glutamic acid decarboxylase that is found almost exclusively in GABAergic neurons, and it is actually found throughout the brain rather than being localized to specific areas or pathways. There are two types of receptors GABA interacts with, GABA A and GABA B receptors. GABA A receptors are ionotropic receptors. When GABA binds to the GABA A receptor, it causes the opening of an associated ion channel that is permeable to the negatively charged ion chloride. When negative chloride ions flow into the neuron, they hyperpolarize the membrane potential of the neuron and make it less likely the neuron will fire an action potential. GABA B receptors are metabotropic or G-protein couple receptors, and when activated, they frequently cause the opening of potassium channels. And these channels allow positively charged potassium ions to flow out of the neuron, again making the neuron hyperpolarized and less likely to fire an action potential. A quick summary of what was covered in this video. In the central nervous system, acetylcholine is primarily excitatory. It plays a role in arousal, memory, learning, and neuroplasticity. The major function of central norepinephrine system is to regulate arousal and modulate reward circuitry. Humans need serotonin to regulate mood, appetite, and sleep, and it also plays role in cognition such as memory and learning. Gamma-aminobutyric acid or GABA functions to reduce neuronal excitability by inhibiting nerve transmission. Glutamate is the most abundant neurotransmitter in the brain and in the CNS, and it is involved in virtually every major excitatory brain function. So it plays a prominent role in neural circuits involved with synaptic plasticity, which is actually the ability for strengthening or weakening of signaling between neurons over time to shape learning and memory. Last but not least, there's dopamine. And dopamine is considered a special type of neurotransmitter because its effects are both excitatory and inhibitory, which effect depends on the type of receptor that dopamine binds to, and of course the brain area where the dopaminergic neurons innervate. So dopaminergic pathways in the brain influences reward process such as wanting and liking and the reinforcement of pleasure behavior. It is involved in executive functioning, for instance, goal-directed behavior, cognitive flexibility, and problem solving. It is also involved in associative learning, which is acquiring and modifying behaviors and skills and such. And last but not least, in motor control, which is the coordination and control of reflexes as well as voluntary movements. What is the role of histamine in the central nervous system? Something for you to ponder upon. If you like this video, hit like, click subscribe, and click on the bell so that you will be notified whenever a new video is released. So let's move on to the next topic, and in the next video we'll be covering the pathophysiology of CNS disorders.